thank you all. So today I am going to talk about plasmonic nanocapsules for photothermal therapy. So before going into the technical details of my work, I will give you a brief introduction about what plasmonic nanoparticles are and how can we use them for photothermal therapy. So before going into the talk, I should say that I am from IIT Mandi, which is located in the state of Himachal Pradesh in the northern India, and this is our south campus, and this is our north campus. Probably this is the only IIT where it snows. So I welcome you all to visit our campus sometime. This is a beautiful campus. So once the pandemic is over, I hope you will make a visit to this campus and we'll have a chance to interact with you all. So coming to my research area, so my group works in the interface of material science and biology. So where we synthesize different kinds of nanomaterial and look towards the application of these nanomaterial for different biomedical applications. So today the focus of my talk would be plasmonic nanomaterials and its application in photothermal therapy. So we should, so before going into the details, we should understand what is a plasmonic nanoparticle. So when we move from a bulk metal to the nano sized metal, so the most intriguing property which we achieve is the surface plasmon resonance. What do we mean by surface plasmon resonance? That if we shine light onto these metal nanostructure, what will happen is that the electric field present in light will cause polarization of the metal nanostructure. What will happen is that the conduction electron will get polarized and we will have a kind of dipole created in the metal nanostructure. So this kind of plasmon resonance we can also create in an extended metal film through some specific configuration where we can have an electron rich and an electron deficit region. And when we shine light of a particular frequency, if it matches with that, the frequency of the oscillation of this electron, we achieve an extended surface plasmon resonance. However, in case of metal nanoparticle, once this dipole is created, since the size of the nanoparticle will be much smaller than that of the wavelength of the light, so what will happen is that as the light changes its direction, as the electric field vector changes its direction, so this dipole will also start oscillating. So this oscillating dipole causes the absorption of the light, and we get to see a color in the metal nanostructure. So, and these this is called the surface plasmon resonance, which is kind of the coupled oscillation of the electron on the surface of metal nanoparticle, which is coupled to the electric field of the electromagnetic radiation, which are impinging on it. So the interesting property is that we can tune the dipole length by changing the shape and size of the nanostructure. We can also create different modes of dipole as well as quadrupole in the nanostructure depending on the shape. So let us look into how shape of the nanostructures affect the surface plasmon resonance. So I hope the surface plasmon resonance phenomena is clear. So if we take a spherical nanoparticles and if we impinge a light of suitable frequency, what will happen is that we can get only one kind of polarization because of the symmetric nature of the particle. And what we get to see is that absorbance happening at a specific wavelength. For example, in case of silver nanoparticle, we get to see an absorbance around 410 nanometer, and like even if we change the size of the nanostructure, the specific at which the surface plasmon resonance is occurring is changing. So what will happen is that if I increase the size of the nanostructure, if I suppose from 40 nanometer, if I make it an 80 nanometer particle, what will happen is that the length of the dipole is changing. So the energy needed to keep the charge separated will be less because the restoring force will decrease, as a result of which what will happen is that the surface plasmon resonance wavelength will shift towards higher wavelength or lesser frequency. So similar to that, if we change the structure, keeping the kind of material constant, if we prepare a nanocube, so what's happening over here is that in case of a nanocube, we are having some sharp corners. So we know that electron cloud loves to stay at the corner, and because of that, what happens is that the length of the dipole is increasing over here. So due to which there is a red shift in the LSPR peak of the nanostructure. So once we move from a spherical 40 nanometer sized nanoparticle to a 40 nanometer cube, so there will be a red shift in the spectra. Again, like if we change the shape for, if we prepare a tetrahedron and an octahedron, where again like we are increasing the corner sharpness, so again the charge will get accumulated at the corner, and further the length of the dipole will increase, leading to a red shift in the spectrum. On the other hand, if we prepare a kind of shell-like nanostructure which will have a core of a dielectric which is coated by a thin metal surface, So in this case, what we achieve is that the dipole, what happens is that the SPR wavelength shifts toward the higher wavelength. So what we conclude from here is that a solid structure, if we take a solid nanostructure, if you play with the structure, we can tune the surface plasmon resonance, and because of that, the absorbance changes and we get to see different color in the nanoparticles in their colloidal form. And when we prepare a hollow nanostructure, in that case also we can shift the SPR peak towards higher wavelength. So similarly, if we prepare a rod-shaped nanoparticle, so we can polarize the nanoparticle in two different ways. Longitudinal axis and on the transverse axis. So you can clearly see from this slide that once the polarization is in the longitudinal axis, so the charge separation is higher, so the restoring force is less, and because of that we get to see a difference in the SPR peak position between the longitudinal and the transverse oscillation. So you get to see that transverse plasmon band occurs at a higher frequency or lower wavelength, and the longitudinal surface plasmon peak happen at a higher wavelength. So the reason behind getting into the details of surface plasmon resonance is that because the kind of nanostructure I am preparing, I'm trying to tune the wavelength of the surface plasmon resonance so that it can be used for photothermal therapy application. Another important property of metal nanostructure is that we can achieve nanoparticle coupling, which can lead to intense electromagnetic field at the junction of the two nanoparticles, where the two nanoparticles lies close to each other. So at the space between these two nanoparticles will have much intense electromagnetic field, which can be exploited for surge-based sensing as well as surge-based bioimaging application. Further, when a frequency of light is impinged on it, which is leading to the emission of plasmon, that will also cause rise in temperature of the nanostructure, because the electron cloud will there will be a phonon electron coupling, which leads to rise in temperature of the nanoparticle. So that can cause rise in the temperature around the surrounding of the nanostructure, and which can be exploited for photothermal therapy application. So this particular slide shows that how impinge a laser of a suitable frequency onto the surface of nanoparticle, and we achieve polarization, and that electron can come with the lattice and an electron phonon coupling, leading to heat generation, and that can be used to killing of cancer cells. As we know that when we prepare a nanoparticle, due to the size of in the nano range, we can achieve passive targeting to the tumor site by exploiting the enhanced permeation and retention effect, or the EPR effect. So even if you are not attaching any specific targeting molecule onto the nanoparticle, still it has the it has a higher probability to accumulate at the tumor site because of the leaky blood vasculature. So this right-hand side shows that if we inject nanoparticle onto it, into the bloodstream, it can get accumulated into the tumor site. And now if the nanostructure is made of suitable shape, and if we shine an NIR light onto it, these nanostructures are going to absorb this NIR light and generate heat, and that heat can be used to kill the cancer cells. And this phenomena is known as photothermal therapy, where we are using light to generate heat, and that heat is leading to the death of the cancer cells. So now what kind of wavelength are we going to choose for this kind of therapy, this phototherapy? So we have to be very careful in selecting the wavelength, because we know that we need to shine light onto the human body. So that light should be absorbed least within the human body, so that it allows the light to penetrate into the tissue site and reach the tumor site, where I will be delivering the nanoparticle, and so that the nanoparticle present there can absorb this light and can generate heat. So looking into the absorption of the different biomolecules and the blood present in the human body, so what is being observed is that, so if we use NIR light, it has a higher penetration depth. It can get into the tissue to up till certain centimeter depth, and that can be used for generating heat if we deliver nanoparticle to that site. So now this biological window is divided into two sections. So the first one is the first biological window, is around 700 to 900 nanometer range, and the second biological window is from 1000 to 1300 nanometer range. So the second biological window is more preferred because of its higher penetration depth. So it is always better to prepare nanostructures which will absorb in the NIR light, and preferably in the second biological window. Now coming to the technical part of my talk. So what we do is that in our lab we prepare some rattle-like nanostructure. I'm sure that you all might have seen the rattles, which is a toy for a kid, which has a solid object caged inside a shell-like structure. So we try to this toy, this rattle toy, and try to prepare a nanoparticle out of it. So how do we do that? So initially, what we do is that first we prepare a gold octahedral solid nanoparticle through following a wet chemical synthesis process, where we use a kind of three-step seed mediated growth process. So once the octahedron is prepared, what we do is that we create a cubic shell around it by using silver precursor and a weak reducing agent, which leads to a kind of formation of cubic shell around this octahedron nanoparticle. And then following a galvanic replacement reaction, what we get to see is that if we add gold salt into these nanostructure, what will happen is that due to difference in the reduction potential of the two metals, Ag and Au, it forms a porous thin around the solid core gold structure. So why we prepare this kind of structure? So the reason behind this is that we are able to shift the surface plasmon resonance from the visual region to the NIR region by creating a porous hollow structure. In addition to that, what we will get is that we will achieve the presence of intrinsic electromagnetic hotspot. As I have mentioned in my previous slide, that if I have two nanoparticles lying close to each other, so the region in between the two will have intense electromagnetic field, and that can be exploited for bioimaging as well as search sensing application. So now here, when you have a solid gold core surrounded by a thin shell of gold, so the junction at which this two surfaces are lying, that will lead to the generation of intense electromagnetic hotspot. Other than that, since being a porous structure, what we can do is that we can load certain therapeutic molecule or a drug molecule inside this structure and can be used for drug delivery application. Here you can see the figure which demonstrates the extinction or the UV visible absorption spectra of the nanoparticle. So the pink color is the core AU octahedron when we silver shell around it. So we see a yellow color because the surface plasmon resonance peak shifts towards the higher wavelength. Now when we go for galvanic replacement reaction, we get to a kind of bluish colored solution, which is due to the presence of this porous hollow nanostructure and have an absorbance in the NIR range. This slide shows the TM image of the nanostructure, where you can see this is the AU at Ag structure. You can clearly see the difference in the contrast between the solid AU core, which is lying inside the cubic shell of the silver. Now, once we perform a galvanic replacement reaction, what we end is we kind of structure where we have a cubic octahedron. So we have a cubic silver porous shell which is caging a solid octahedron inside it, and this is the right image of that. So these regions, porous regions, and also the region of intense electromagnetic hotspot, which can be used for several applications. So now what we did is that after gold metals, we loaded this nanorattle with a Raman reporter molecule, which is benzene in present case, and this benzene diethyl is into the structure. And after this, what we did, we thin solid silica shell around this nanorattle structure, so that the BDT molecule which is lying in the cage of the gold nanorattle will not come out. Now further what we did, we performed an additional growth of mesoporous silica nanoparticle around it, and we used the porous silica nanoparticle for loading the drug doxorubicin inside it, as a result of which this entire nano-composite can be used for simultaneously SIRS-based bioimaging as well as fluorescence bioimaging, of doxorubicin, which is intrinsically fluorescence in nature, as well as it can act as a drug carrier, where we are demonstrating using a chemotherapeutic drug doxorubicin in the present case. So this is the TM image of the nanostructure. You can see the nanocore nanorattle line inside the solid shell as well as the porous shell of the silica nanoparticle. And this particular slide shows the SIRS performance as well as the fluorescence performance of the nanostructure. Generally, it is observed that whenever a fluorophore is present in the vicinity of a metal nanostructure, it quenches. But due to the presence of a thin silica, solid silica shell, the metal nanoparticle which is in the core do not quench the fluorescence of the doxorubicin, which is loaded in the porous shell. Further, the solid silica shell also protects the BDT molecule which is loaded in the core and not let it come out, and we are able to perform search imaging through that as well. So this particular slide shows the search imaging of a CF7 cell. After delivering the nanoparticle to it, we perform a search mapping of the cell, and we can see that the nanoparticles are taken up by the cell, and we can see the presence of these signals arising because of the presence of BDT, which is present in the core of the nanoparticle. And the right-hand side image shows that the doxorubicin, which is loaded inside the cell, is also can be traced using confocal microscopy. And independent manner, if we move from 1 hour to 4 hours, we see an increase in fluorescence of the doxorubicin molecule, which is clearly demonstrating the uptake of the nanoparticle as well as the release of the doxorubicin drug inside the cells. Next, we evaluated the therapeutic potential of this nanostructure compared to the non-loaded nanoparticle. With that of loaded particle, we can see a significant decrease in the cell viability with increase in concentration of the nanoparticle. And we also performed we tried to understand the mechanism of cell death. So it is quite established that the way doxorubicin kills. So here we have seen the generation of reactive oxygen species, as well as like we have performed certain apoptotic assay. We can clearly through AOEV staining we see the control cell and the treated cells, where we can see formation of the penetration of the ethidium bromide dye in, because of the compromised cell membrane. And also here, by lighting the nucleus of the cells, we can see a chromatin condensation in the treated sample as well as fragmentation. And here the figure C shows the SEM image of normal cell and a treated cell. We can see the formation of apoptotic body in case of the treated cells, demonstrating that the nanoparticle loaded with the drug induced apoptosis in the cells, leading to cell death. So now we thought that once we prepare this nanostructure, can we use it for a stimuli responsive therapy? So here what it is that we use a phase change material, which is a tetradecanal molecule and which stays in a waxy solid state at normal temperature, and if the temperature rises, it becomes it gets transformed into a liquid state, and it can if we load the tetradecanal molecule, porous structure of this nanorattle along with drug molecule, and if we shine light onto it, what will happen is that due to the heat generated because of the nanostructure, that will melt the tetradecanal molecule and release the drug molecule from inside the cell. So we achieve a stimuli responsive therapy. So this is the SEM image of the nanostructure, and we also perform search mapping through it. And this particular slide shows the photothermal performance of the nanostructure. If you shine light, we can clearly see a rise in temperature, and it is also photostable. Like for several rounds of cycle, the rise in temperature remains constant. And then we use this structure for drug delivery and photothermal therapy application. We can clearly see concentration at 200 mg per ml concentration. When we have both doxorubicin and laser impinged onto it, the cell death percentage is much higher than that of the nanoparticle just loaded with doxorubicin without any laser treatment, which clearly demonstrates the stimuli responsive chemotherapeutic potential as well as photothermal therapy potential of the nanostructure. So this summarizes the entire thing of the nanorattle being delivered to cells for stimuli responsive therapy. So further, we thought that can we prepare a nanoparticle which have absorbance in the second biological window. So in that case, what we did is that we prepared an elongated capsular structure having a solid gold core and a rod-shaped porous gold shell, which will have in the NIR2 region as well. We followed the same process of generating template. We 5 different sizes of nanoparticles, which is AV over here, and then perform galvanic replacement reaction of this template and 5 different sizes of nanocapsule, which is again a rattle-like structure having a solid gold core and a porous shell. So this is the SEM image of the nanocapsule at a partial galvanic replacement, and this is after complete galvanic replacement. You can clearly see the structure over here, which can be used for loading of therapeutic molecule, and also it gives rise to absorbance in the NIR range and can be used for photothermal therapy application. So this slide shows the if we increase the increase in laser power and with concentration, we can clearly see a rise in temperature, and here you can clearly see the colloidal solution of the nanostructure at different concentration captured through a NIR camera, and we see an increase in the the temperature rises with increase in concentration of the nanoparticle. So this is the data of photothermal therapy of using this nanoparticle. Here what we did is that we used the nanocapsule, we conjugated the surface of the nanocapsule with the Herceptin molecule, which is a targeting agent for breast cancer cells. So and we tried to study the stimuli responsive therapy using a targeted approach. So what we see that when we use 100 mg of EU nanocapsular structure at a very low power density of 0.5 watt per centimeter, almost 100% cell death in case of Herceptin conjugated gold nanocapsule compared to other structure. So this nano, again, like as I mentioned, this has an intrinsic electromagnetic hotspot. It can be used for SIRS application. This slide shows the SIRS performance of these nanostructure, where the set one, which is the smallest size of nanostructure, having the best SIRS performance and can be used for SIRS imaging. This is the FDTT simulation data clearly demonstrating the intense electromagnetic field around the nanostructure. And again, we use it for some sensing applications, so I'm skipping this. So the summary is that we prepared some plasmonic rattle-like nanocapsules which have absorbance in the NIR range and which can suitably be used for photothermal therapy application. It has a porous structure which can be used for loading drug molecule, and the intrinsic electromagnetic hotspot present can be used for SIRS sensing as well as SIRS bioimaging application. So with this, I would like to thank my group who has contributed towards this work, and to the funding agencies for their generous funding. Thank you very much.
Thank you very much, Amit. There are we have time for just 2 questions and you have to be brief. So the first question is from Dinesh Kumar, who wants to know if there are any preclinical studies which have been performed with these nanosystems or nanocapsules that you have. So can you be very brief?
Yeah, sure. So presently in our campus, we do not have any animal house, so we were not able to carry out the preclinical study, but I am collaborating with other institutes. So hopefully soon we will be performing some preclinical study using these nanostructures.
Okay, thank you. And there's another question. I'm not sure from whom, whether the nanoparticles move inside the shell.
Okay, so yeah, in case of a rattle, we see that the solid object inside it moves, but the nanoparticles we prepared, though we can tune it to make it move, but the structures which we have prepared in our lab, the solid object lying in the core does not move.
Okay, I have one question, and then how do you ensure that your nanocapsules are targeted to particular sites, or how do you target them to particular sites?
Yeah. So thank you, ma'am. So for targeting, what we do is that initially we coat this nanostructure with a bifunctional peg which has a COH group and a thiol group. So thiol will attach to the metal surface, and we have a free CO- group which can be attached to some ligand which can specifically target some specific cancer cell types. So we exploit this COH and we can conjugate it with some antibody. So in present case, we have used trastuzumab or Herceptin molecule, which is specific for breast cancer cell. So we can do conjugation of different other targeting molecules as well.
Okay. Thank you very much for a fascinating talk. Thank you.